With pleasure that I introduce Dr. Bruce Mathieson, who's an Associate Professor of Biology in the Irving K. Barber School of Arts and Sciences. He will speak about the Nobel Prize in Physiology or Medicine. This year, the prize goes to Yoshinoro Ohsumi for his discoveries of mechanisms of autophagy. Dr. Mathieson.
Thank you. Thank you very much. Welcome. This winner of Physiology and Medicine, Dr. Ohsumi, he is a Japanese researcher, and he's a very humble man from what I understand, and he began working on this species here, which some of you probably recognize in the audience. It's the wine yeast, the little creature that you put in grape juice and it turns it into wine. So they're beautiful cells. I think they're beautiful cells, and inside those cells, it was discovered before Dr. Ohsumi started working on it, a process called autophagy is at work. And apparently, during the 1990s, when he and his lab mates worked on this, they discovered that this process is not just present in yeast cells, but in all of our cells, all the way to the most complicated cells. And what those little bumps are, I've learned, is places where when they reproduce, they leave that little scar behind. And I was fascinated by that, but it actually has nothing to do with autophagy. They started studying this when they found that if they take yeast cells in culture and they starve them of nutrients, the lack of nutrients caused the cells to change. And inside, the cells filled with this structure called an autophagosome, which is a membrane-bound structure. And what they discovered later, what's going on, is that the cell is actually quarantining unnecessary proteins and lipids and sugars into this structure and breaking them down enzymatically and then reusing the little components for what they do need to keep alive because they've been starved. So many years later, we know that this process of autophagy is not only important for yeast, but it's also important for health, and many diseases have been identified that are linked to this process when it goes wrong, because we need to have it to happen for lots of different circumstances. So this is a picture of the inside of a cell. There's a cell membrane. This is outside the cell. This is inside the cell. It's a cytoplasm inside the cell. It's a very dynamic place. Not only is the cell manufacturing new proteins and new lipids and things, but the older proteins, et cetera, they have to be recycled when they get old. If you don't remove the garbage, so to speak, the cell fills up with toxic levels of non-functional structures. And that can cause disease conditions, which I'm going to show you two examples of. Here's a picture of, talk about molecular motors. This, I think, is a good example of a molecular motor, because out of nothing, these cells, when there is something that needs to be recycled, there's enzymes that Dr. Ohsumi discovered, 16 different genes, is under very good control, tight control. They build a membrane that surrounds the structures and sequesters it into a little sphere. And then the enzymes that are necessary for degrading it are put inside that sphere, and it breaks them all down like a little machine. So there's a picture of the membrane forming around all these structures. You can see it's called the autophagosome, and it's destroyed, the stuff in the middle, because of this structure here that binds to it. It's called the lysosome. It's filled with hydrochloric acid and acid-loving enzymes called the hydrolases that destroy all of the stuff that's in here, the garbage. So when this thing fuses, this vesicle that forms degrades everything inside, and it gets removed. And that's in the simplest cell, like a yeast cell, and in cells like your and our cells, even our neurons, the most complicated neurons or cells that we can find. Now, we need to recycle this bad stuff when it gets old all the time. Just the act of living causes old structures that need to be removed. Okay, and this is one of the organelles. In fact, it's not just proteins, but little organelles that are in the cytoplasm. This is a mitochondrion, which is what takes fuel that we eat and strips away the electrons, delivers them to oxygen, and it creates energy. Well, that's a good thing, except that mitochondria produce very toxic free radical structures, which are very reactive free electron chemicals. And they can be very toxic. They can actually destroy the mitochondria itself. They can destroy the DNA in the cell. They're very bad. And older mitochondria produce lots of these free radicals. So we have to make sure we clean away the old mitochondria before they start doing that, because it will kill the cells. And you can see what happens. Here's an autophagosome that's depicted here with one of those mitochondria, and there's a lysosome that comes in and fuses with it, and all of the stuff that's inside gets degraded, and we get rid of the toxin-producing old mitochondria before it causes damage. Now, the first example of a disease, I'll say, is Parkinson's disease. And in Parkinson's disease, most of you probably know, or you've heard of this, that there's a type of neuron called a dopaminergic neuron. It synthesizes dopamine neurotransmitter, and it's associated with problems of movement. So this is part of the brain. This is the midbrain of a patient with Parkinson's, and you can see actually a normal brain. This is the normal brain. Dopamine is a dark staining molecule, and you can see the millions of neurons in there give it this little dark hue. They call it the substantia nigra because of that dark hue. In a Parkinson's patient, almost none of that darkness is there. The dopamine neurons have been killed, and it turns out now we recognize that mitochondria have not been removed by autophagy as they get old, and they create too much of this free radicals, and it kills those cells. There's two genes that were long associated with the familial or inherited form of Parkinson's disease, PINK1 and Parkin genes. We didn't know what they were doing. We just know they were associated with this disease. But now we've identified, just in the last 5 years, that those two genes code for proteins, PINK1 and Parkin protein, and those regulate the autophagy process of mitochondria before they get too old to be functional. Okay, so if autophagy is dysfunctional, is it something wrong with autophagy, those old mitochondria stay in the cell and they kill the cell and the dopamine-containing cells because of the free radicals they produce. So that's the first example. Second example is autism spectrum disorder, or ASD. It's a fascinating disease in some respects. People have been studying it, and lots of people know autistic children and families, and they recognize now that in autism spectrum disorder, in some forms of this, there are actually too many neurons and too many synapses, that is, connections between neurons, that have formed. And in fact, less than 5 years ago, a study came out that showed that in our first year of life, we have an incredible proliferation of new neurons making new synaptic connections. This is an illustration of a neuron in the cerebral cortex, and each of these little threads coming off is a part of the cell, the neuron. That's where the synapses, the connections with other neurons, take place. If you take one of those little dendritic branches and look at it in a higher magnification, every one of those little bumps there is a place where a synapse is allowed to be created. The more of those bumps, the more synapses. So from what I told you, they've learned that early on, we have way more synapses than we need. We have to remove all those little bumps in the synapses to make a normal developmental maturation take place. In this study, they compared brains of normal age-related children as they grew versus autism spectrum disorder children. And they can see here there's a control group here, the normal development. Each of these little dots is a neuron, and they just counted how many bumps are on the dendrites. So you can see that they start off in these ages, 1, 2, 3, 6, 8, 13. They've lined them all up in an age developmental series. We start off with lots of synapses, and then around 13 to 16, they drop by half. We've lost half of the synapses in a normal child. Autism spectrum disorder, they start off a little higher than the normal controls, and it's called pruning. They don't prune away the synapses correctly. It's either too slow or too non-effective. And the child is left with this confusing cacophony of information floating around, and it's a problem. So this was a really important paper, I think. It illustrates how important autophagy is in a basic developmental process of the brain. Okay, so as a summary, these are a list of different disease conditions or related conditions that they've identified with autophagy dysfunction. And when it's not working correctly, neurodegenerative disease like Parkinson's or developmental delays like autism can form. Immune system requires autophagy for removal of bacteria and fungal infections, etc. And in fact, cancer is also related now to autophagy mechanisms. So 1990s, Dr. Sumi was merely describing yeast to make wine and protecting the cell. But now we know that this is a much, much bigger phenomenon, and it has huge ramifications for health and disease. So thank you, Dr. Asumi, for his work.